Good job. Yeah, that's what I wanted to guys to conclude, that the eukaryotic, so people said shape. Yeah, the eukaryotic is a stick. The chromosomes are sticks, versus the prokaryotic, they're round. Like, so not perfectly round, but they are, they're flexible, but they're a circle, they're a ring. Whereas the eukaryotic is linear. And then another difference is in the size. Like, okay, this is 250 million, and even the shortest one is 47 million. So let's take the shortest human chromosome and compare to the bacterial genome. And here's 1 million base pairs, much smaller. So that's why they don't have time for commercials, right? They don't have introns. Anyway, they are more, the genome is smaller, more concise, and even the distance between genes. So each of these things actually have a meaning, but the distance between genes in prokaryotes is much shorter than the distance between genes for eukaryotes. So let's pretend that I'm doing this in scale. There would be like a gene here, and there would be like another gene over here. In reality, since they're so zoomed out, they would seem so near. But just for purpose of teaching, maybe there's one here and then another one down here. Whereas for bacteria, they would be like right next to each other. There isn't a lot of intergenic DNA. So those are the two differences that, from looking at them, you could tell is that, okay, eukaryotic is not, it's linear, and prokaryotic is circular, and the size of the genome. And then you might wonder, but our mitochondrial genome is circular. Just out of curiosity, do you guys know of the origin of mitochondria? Why would you think that we have circular genomes? But then I typed the question, why are eukaryotic mitochondrial genomes circular? Yes, so I see West probably learned this in another class, or you just came up with a very good theory, which people have come up with. Yes, so the prokaryotic cells, they got engulfed by another cell, and now they are part of that other cell. So what we think is that an eukaryotic cell ate up prokaryote, and then the DNA of the prokaryote became part of that eukaryotic cell. So this guy here, its origin is from prokaryote, and so that's why it has a circular shape, because in origin it was a prokaryote, but now it's part of the eukaryote. And if you keep studying these things, you're going to learn that some of the genes that were in the mitochondria got shipped to the nucleus, but that's just like fun stuff that I'm telling you out of curiosity. All right, so I alluded to DNA that's coding versus non-coding, DNA that are genes versus not genes. So remember, the gene is the DNA that is transcribed. So it's that DNA that the DNA still exists as DNA, but there is now a molecule that is an RNA molecule that was made based on that DNA information. And sometimes that RNA molecule is translated, sometimes not. So the parts that are not translated, they're called non-coding. So when you think of coding, think this is some sequence that are a code for another sequence. So it's an RNA sequence that are a code, so the codons, the triplets of RNA, they're going to encode an amino acid. If they're not encoding an amino acid, they're non-coding. So here you have a DNA molecule, and it still exists as DNA in the cell. Whenever we say DNA goes to RNA, we're just being lazy. It's just that the RNA polymerase makes this other molecule based on information it reads from here, or either this strand or this strand. And then the ribosome will read the RNA and come up with the protein. So the ribosome will read the RNA, and there will be the tRNAs that bring the amino acids. They'll build a protein. So that's translation. But that doesn't always happen. So there are molecules that are active as RNA. Here are examples, tRNA and the ribosome. And there's something that you might hear in other classes, it's called RNAi. This just means that sometimes you make RNA that is an mRNA, but that mRNA gets degraded before it becomes protein. Or, so I'll just stop there. So not every RNA becomes protein, either because it's not meant to become protein, it's just meant as functional molecule as a tRNA or a ribosome, so rRNA, or it's an mRNA that gets destroyed before it becomes protein. You could think about it also as the intron of a gene that is transcribed as mRNA but doesn't get translated to an amino acid. So here's an example of the non-coding DNA. It's a stuff that gets transcribed, not translated. And okay, I think I went over this a few times, so I'm just going to keep going from this one. And I mentioned earlier that when I say genomic genes, genomic DNA, I ask, does genomic mean that it's DNA that has genes? I wanted to know it's yes, genes, but also non-genes, and actually more non-genes than genes. So this would be our intergenic regions. So again, we have an example of these 3 genes. There's intergenic region. This whole thing is called genomic DNA. And whenever people think genomic DNA, they are not thinking mitochondrial. It's for some reason when they think genomic DNA means nuclear. So it's all of it, the genes and the non-genes. All right, so I'm going to make a poll now with that question. Let's see what you think.